Hello again, Year 13s. Welcome to another installment in our uh, marker-assisted selection. And specifically, when we're looking at marker-assisted selection for selective breeding, how do they actually sequence the genome to know where the differences are so that they can choose the right allele of the phenotype that they are looking for? Okay, so genotyping is a process that determines the differences in an individual's genetic makeup by analyzing their DNA sequence. So my DNA sequence will be different to somebody with dark hair because I have light hair, and consequently that's a different allele, and it will show up differently in our total genotype. Okay, so the guy who invented this was called Frederick Sanger in 1977, and he developed the Sanger method of DNA sequencing. He was a British biochemist, and he worked obviously with a team, and he was awarded the Nobel Prize for this. So what we do, maybe I need to first just start with the definition of DNA sequencing. This is the most direct method by sequencing the DNA of multiple individuals that researchers can identify variations, okay? And that variation, if it holds the trait you're looking for, then it's the QTL, the quantitative trait loci, at specific nucleotide positions. Now, can you remember from the previous video, the BLAD mutation was on chromosome 1 in the gene for, the gene in the position of 383 or something. So it is extremely specific. It goes down to individual nucleotide and their number on this sequence. Then we also have next generation of sequencing, which is the most effective way. And at the end of this, I'm going to ask you to go back to the actual presentation where the video link is inserted to a good video that will take you through how the next generation sequencing works on YouTube. Okay, so the Sanger method of DNA sequencing, what they do to make this work is they use chemically modified nucleotides. So it's like the adenosine, thymine, guanine, and cytosine that you know, but they have been chemically modified so that the base have a fluorescent color. This means if we put them under ultraviolet light, or we shine an ultraviolet laser at them, they glow back in a specific color. And if we do that and we shine on a base, we can actually then see, oh, it's shining back in this color. Therefore, it is this specific nucleotide A, T, G, or C. But because they are so small, we need to make sure that there's only one of these shiny guys in your sequence of your DNA at the specific spot that you're looking for. And we'll get into more detail on the next slide. Okay. However, these chemically modified nucleotides are called di-deoxynucleotides. Okay. I don't think it's that important that you need to know that word. You can refer to them as DDNTPs. So what you need to know is that they shine when you shine a laser on them, give off light, and then the other really, really important thing is when they bind, in other words, when DNA polymerase puts them in the chain that is your DNA sequence, the whole process stops, because DNA polymerase cannot add another nucleotide after a di-deoxynucleotide. Okay, and this process then develops complementary DNA fragments of varying lengths. So let's look a little bit closer at that. So here's your di-deoxynucleotide. Okay, as you can see, di-deoxyribonucleoside triphosphate. So it's got a triphosphate base on it. And the normal one has got this, we call that a hydroxyl group, OH. And the base does not glow like this one glows. Okay. Now, what I imagine he's a Prof. Sanger or a Dr. Sanger, or let's just call him Frederick. What Frederick Sanger did is that he, instead of having a hydroxyl group there, they just put a hydrogen group there. And with this hydrogen group, now the DNA polymerase cannot bind the next nucleotide in the chain onto it, because this is the direction in which it grows is always from the 5' to the 3' direction. So it has to bind there. And because it's got the wrong chemical there, it cannot physically bind onto it. So this is the one, in this case, the base is going to glow yellow. That might be adenine. And then they make it blue for thymine or so forth. So let's see what the next slide holds. So in this case, this is a traditional method. It is very time consuming. And imagine if you have a gene that is thousands of base pairs long, then you have to have thousands of reactions. So you will have 4 different vials, okay? And in these 4 different vials, in every single one of them, you've got normal nucleotides, okay? So the normal thymine, the normal adenosine, the normal guanine, the normal cytosine, they're all in there, and in there, and in there, and in there. And in every single one of them, there's also primers, lots of primers. So the primers is in there, and there, and there, and there. So if we just got the reaction started by heating it up to separate the DNA strands that we're going to add in there, so that the template that we're going to build on, this original DNA sequence. So if we start the reaction with just those normal ones in there, then it will simply be a PCR reaction. Okay. However, what we are going to do is we are going to then add in a low concentration modified thymines, in other words DDNTPs, but it's only going to be the thymine in that one, and the color that it's going to fluoresce is purple. In this one, we're going to have modified adenosines, and the color that it will fluoresce is blue. In this one, in a low concentration, is going to be the color that it's going to fluoresce is yellow. And for cytosine, it's going to fluoresce in green. All right, now we run the whole thing. So if, for instance, we now take the reactions, we run them, heat it up to 98, and then bring it down and to get DNA polymerase going, and heat it up again, bring it down, heat it up again, bring it down, heat it up again, bring it down, as we do in PCR. Then we take the thymine vial and we run that on our gel. And then we take, it's going to go from the bottom. No, it's going to go from the top to the bottom, because remember, the smallest one will always go the furthest. And then the adenosine is there, the guanine is there, and the cytosine is there. So from this, we can learn that the smallest fragment created, when we shone the laser on it, it showed that it had thymine on it. And then if you have a very sensitive ladder that will show you the number of base pairs, you can then say, okay, fine, this is going to be at 1 base pair length. And they have developed it in such a way that it is accurate enough to have a simple single base pair can pick it up. So we know that thymine is now in position 1. However, in that vial, DNA polymerase started in this vial. Wait, let's just go to the next thymine for now. You're going to wonder now, how can it be that it can be so long to actually get the thymine to bind? So what's happening is, because the modified thymine, the DDNTP, is in this vial, but there's lots of normal thymine in there too, chances are that it's going to build with normal thymine, and every now and again it will put down that one that is a DDNTP that fluoresces and then stops the reaction. So in this vial we will have 2 that will be created, but it might be multiple ones. Okay, the cytosine came, we can see that it was then in the second vial. So we had a thymine and then the cytosine. Then in the third one we had a thymine, a cytosine, and then a guanine, and it's in the third position, and so forth. So you can see, if I come back to this one, it's going to be a modified the DDNTP adenosine. However, you started with a thymine, then a cytosine, then a guanine, then a non-modified adenosine, another non-modified adenosine. But can you see down here, the first one it took was a modified one, and the second one it took in this one was a modified one. But that is completely random because we have a low concentration of the modified adenosines. Okay, so as it runs, it will keep on building until it places one that fluoresces, and then it stops. Builds until it fluoresces, and then it stops. And because we then have the different lanes for them, we can then say, when we compare it to the actual ladder, what the number of base pair. So we will know that on base pair number 1, 2, 3, 4, 5, 6, 7 is a thymine. So let's count them. 1, 2, 3, 4, 5, 6, 7. Okay, and this guanine is on base pair number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's see if we're right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, I hope this makes sense. So let's then move on. We then made it a bit simpler and more modern. And instead of using a traditional gel, like in the previous, we now use a capillary gel, which is a very thin tube that is filled with a gel. And as the electricity is applied, the molecules will flow down. And as it flows down, the first one that will pass the laser is going to be the shortest. And the last one that will pass is going to be the biggest. So as it goes past, this detector is basically a camera. And as it goes past, it gives off light. And that light is then detected. And it's detected from the machine, which is captured by a computer. And if you look here, the first one was red. Okay. And if we take that back to here, then we can see that that red was thymine. Okay. DD-thymine triphosphate. And this is dideoxycytosine triphosphate. Okay. And then the second one was purple. Okay, so purple means guanine. The third one was purple, which means guanine. The fourth one was thymine, which is that one. The fifth one was this blue one, which means cytosine. So as the molecules flow by, because of their size, the laser will pick up the light. You can see there as it goes, depending on. So this was the shortest one. I see they ignored that first peak, but you can see that it's purple, purple, red, blue, purple, purple, red, blue, green, red, green, green, red, green, and then we can actually determine how long they were. Okay, I hope this helps. You can also see that the primer makes up this first part. Okay, so that's why it looks weird. The first one is part of the primer, and that's your DNA polymerase that then stops building there. I hope that makes sense. Um, this next generation sequence video, I need you to go and watch. How do I get this thing to stop? Help! Okay, so I need you to go and watch this. Otherwise, YouTube will ping me for copyright infringement. Please open the PowerPoint and follow the link. I really hope this helps. That you understand how DNA sequencing works. Now, you've got the sequence. So you've got all these A's and T's and G's all written out, and you know the sequence. How do you know if your sequence is actually a gene? And the way that we know that is called, we do one of these four things. Firstly, an open reading frame. So an open reading frame is a continuous stretch of nucleotides that begins with a start codon. Okay? The start codon is always adenine, uracil, and guanine. So I said uracil, not T, because in RNA, the T is replaced by a U. Okay? So if it starts with an A, U, G, then you know it's a start codon, and then you follow it all along and you read and you read and you read until you get either a UAA, a UAG, or a UGA. and those are all stop codons. So I just want to remind you the genetic code, and you can see here ATG or AUG, depending on if you're working with DNA or RNA, is methionine, and that is the start codon always. And the stop codon is going to be UAA or TAA, UAG or TAG, and TGA or UGA. So if you read your sequence and you find one of the stop codons, then you read backwards and 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 backwards until you get the start codon. And if you get a start codon and then you get another start codon, then you go back to the previous start codon, if you understand what I mean. Because it will start at a position and then stop at the stop codon. And that's going to show you that the code that you have, that sequence is a gene. Another way is by looking at the promoter regions. So when we have to turn a gene on or off, so you don't always want to express the same proteins, that is usually done by the promoter region. And in that promoter region is usually what is called a Tata box. And believe it or not, it actually has TATA in it. Okay, so the Tata box is recognized by RNA polymerase to start transcription in the promoter range. So over here we're looking at the gene. Over here we're looking at the promoter area. So a little bit before the starting codon. You will have the promoter box, and that is the Tata box. Another way is looking at the transcription, which is when RNA polymerase copies it into RNA from the DNA that's in the nucleus. So we take the DNA in the nucleus, RNA polymerase comes and reads it because the DNA never leaves the nucleus. And then it transcribes it into RNA, and then it takes that RNA and takes it out of the nucleus. Now usually there's some processing going on, and that means we take the unwanted parts out of the gene, and we call those introns. We get rid of them, and then they also add a 5' cap and a 3' cap, getting rid. So basically it's copied, and then you have a version of the copied one that was refined. Okay, so if you have that, that is another way of looking at it. Then another one is conservation across species. So let's say you found the black hair gene in mice, and then if you find a gene in cattle that is the same as the black hair gene for mice, then you can say, oh, okay, it's the same as that. And because they might have shared a ancestor many thousands of years ago, that gene might have remained the same. So if you think about us and chimps, there's many genes that's going to be similar. The same with us and many other mammals. Okay, so we share a lot of genes. And then lastly is the functional studies, which I can't go back to now. Okay, I hope that helped. The functional studies, you just simply take the gene or the DNA, and then you either delete the gene and see what happens to the organism, or actually sees what happens to the organism when you express the gene. So you can actually make RNA, put it into a cell, and then see what happens. All right, so that's your functional studies, either by taking it away or artificially inducing it. I hope this helps and that you understand DNA sequencing. Bye.